Welcome viewers. Today I am recording another topic regarding homeostasis in plants. You see, plants which are sessile, do not move, need to maintain temperature homeostasis. Every biological organisms regulate themselves to a relatively stable state. Plants apply different mechanisms to maintain homeostasis. They are sorry they are role of stomata, regulation of temperature, regulation of water and food materials, guttation and homeostasis, removal of other metabolic waste, osmotic adjustment, and homeostasis at organellar and molecular level. First, role of stomata. When light falls on stomata, we see that from epidermal cells, the potassium ions enter inside the guard cells. Along with the potassium ions, water molecules also float into the guard cells. As a result, guard cells turgid or swellens up and stomata is open. Just the reverse happens when the light goes out or where there is a night dark. The guard cells shrunkens and flaccets. Thus, stomata are closed. Stomata exchange oxygen and carbon dioxide with atmosphere. Stomata transports water and minerals. Stomata regulates internal body temperature. Next, we come to regulation of temperature. This is a plant called desert brittle bus, Encelia farinosa, scientific name. This plant shows an adaptation having hairy leaves that lowers the loss of water to evaporation and catches water from the air to keep itself alive. Similarly, some of the deciduous trees, they drop their leaves in cold weather to reduce their metabolism. Next, regulation of water and food materials. We see generally plants built in order to regulate its water use. CAM plants, known as granulation acid metabolism plants, absorb carbon dioxide during night when the stomata are open and convert it to C4 compounds, which are oxaloacetic acid, malic acid, and aspartic acid, and utilize it during daytime to produce glucose in C3 cycle when the stomata are closed. Cactus have spines in place of leaves to reduce transpiration. Opuntia have thick stems called phylloclades that can store water for a long time. Mangroves are adapted to saline environment and can secrete sodium through their leaves. Next, guttation and homeostasis. At night, transpiration does not occur because most plants have their stomata closed. If there is a high water content in soil, water enters the roots and is accumulated in xylem vessels. Some plants, such as grasses, force this water through special pores known as hydathodes, present at leaf tips, edges, and form drops of liquid water. This process is called guttation. Removal of other metabolic waste. Calcium oxalate is deposited in form of crystals in the leaves. These trees shed their leaves early. Resins, gum, latex, mucilage are such kind of waste which are excreted by plant materials. Next is the osmotic adjustment in plants. Hydrophytes have broad leaves with a large number of stomata on their upper surfaces. This helps them to remove extra amount of water. Xerophytes have thick waxy cuticle over their epidermis to reduce water loss. Halophytes absorb more salt along with water. Water tend to move out of their cells into the hypertonic if it enters, water tends to move out of their cells if the cells are placed in the hypertonic salt water or any kind of solution. But this does not happen. Why? Because when salt enters into the cells, plants carry out active transport to move and hold large amount of salts in their vacuoles. Salts are not allowed to move out through the tonoplast of vacuoles, as we can see in this picture. This is a cell. Here is the tonoplast, the outermost covering of vacuole. NaCl, after entering inside the cell, it is forced inside the vacuole where it is stored. The high salt concentration is maintained inside the vacuole and it is not allowed to move out of the tonoplast into the cell, thereby maintaining the homeostasis of the cell. Homeostasis at organellar and molecular level. Glutathione homeostasis. Level of glutathione, or we call it in short form GSH, tends to alter in relation to temperature and other biotic and abiotic stresses. Then mitochondrial homeostasis, redox reaction that occurs in the mitochondria, they also help in maintaining homeostasis. Homeostasis in Arabidopsis. In wild Arabidopsis, a constant size is maintained. That is by the help of bonsai 1 gene. This particular gene helps to regulate cell expansion and division. Aquaporins and water homeostasis. Arabidopsis is found to express 30 genes. Those genes control vacuolar and plasma membrane aquaporins, which are responsible for cytosolic osmoregulation. Next, auxin homeostasis. Auxin, a phytohormone, links plant growth with stress adaptation responses through salicylic acid and abscisic acid signals. Next, copper, iron and zinc homeostasis. Iron, copper and zinc are metal activators for numerous enzymes. They are toxic at higher levels. Organisms maintain cytoplasmic metal concentration, which is known as metal homeostasis. Metal responsive transcription factors regulate trace metal metabolism and are able to sense changes in metal concentration and coordinate expression of genes and coordinate the expression of genes that are involved in distribution and use of metals in the cells. Next, peptides and plant homeostasis. Plants' natriuretic peptides (PNPs) modulate ion channels and water uptake and maintain homeostasis. Phytosulfokines (PSKs) and rapid alkalinization factors RALFS have roles in development and defense, also have functions in plant homeostasis. Nitrogen, phosphorus and sulfur homeostasis. Key root membrane transport Proteins and molecular regulators control root uptake systems in response to nitrogen, phosphorus, and sulfur availability. This is known as nutrient homeostasis. The last one, a noble intracellular potassium hydrogen antipoda. Sodium hydrogen antipoda is important for potassium ion homeostasis found in tomato. So this was all about the concept regarding the topic we discussed today, the homeostasis in the plants. Well, thank you, and we will see the next topic in the next video. Till then, take care and thank you.